hope you're all nicely hydrated. I feel I should keep things moving so we keep somewhat on track. Just a short apology. Unfortunately, Dr. Stephen Van Leeuwen was unable to make it today. So you have a fill-in, last-minute fill-in. I can't promise the quality of the talk, but apparently they're very attractive. Anyway, I will present on some work that I did. Some of this work started when I was in New Zealand recently, and it's a bit of a collaboration with some colleagues, so I'll just jump into it. So my talk today is on Bayesian analysis to identify traits associated with the invasiveness of Phytophthora species. And excellent. So welcome, thank you. My name is Peter Scott. Today I'll be talking about Bayesian analysis to identify traits associated with invasiveness of Phytophthora species. And that might sound a little bit off topic, but I think this is actually very relevant to the direct management of Phytophthora diseases and other diseases in general. So I'll talk briefly about what we did, but first I'd just like to acknowledge the contributors. So this work, I have been directly involved in this work for some time. I was involved in the experimental design and getting a lot of the data. My colleague Trina was also very helpful in collecting data for this work. And for this particular project, a lot of the disease modeling was by a colleague from Bruce Marcotte from the USDA in Oregon, who was incredibly generous with his time and support in doing the disease modeling for this particular project. Oh, I would also like to acknowledge my colleagues in New Zealand Forest Research, and the New Zealand Better Border Biosecurity, because they were very instrumental in this work, especially in the early years. So I feel every time I do a talk about Phytophthora, I have to have the standard images of doom and death. So ready yourself for some death. So briefly, we all know some Phytophthora species cause very serious diseases globally. And I think probably one of the best examples of that is the infamous Phytophthora cinnamomi. In Western Australia here, we've got an image of it being associated with massive dieback in our forest, which is very tragic. But this Phytophthora cinnamomi is also a really nasty beast globally. And here's an image of it causing oak dieback in Sardinia. And, you know, it's very well acknowledged as one of the most significant global plant pathogens on the planet. And there are some other really nasty Phytophthora pathogens. We've had lots of talks today from Sarah and others talking about Phytophthora amorum. This is an aerial Phytophthora. They call it the snake that can fly. It spreads through the air. It infects many, many different trees, causes horrible bleeding cankers, and yeah, it's very upsetting. Some other Phytophthora seem to be limited in their impact. Now, these Phytophthora might be very important pathogens that cause a lot of disease, but currently there are some Phytophthora that don't appear to cause major problems. You know, and this sort of makes sense. Everything's a gradation in biology. Some Phytophthora pathogens probably have a limited host range. Some of them are like Phytophthora cinnamomi or Phytophthora amorum. You know, and it's really hard to work out where these Phytophthora fit on this spectrum. So this is an example. This is Phytophthora aleatoria, which is a Phytophthora that's localized largely to parts of the South Island of New Zealand and in nurseries throughout New Zealand. But at the moment, it appears only to affect pines, and it only seems to cause disease under very, very specific environmental conditions. Here's another really interesting example. This is Phytophthora canovii. This is a Phytophthora we think is probably native to New Zealand. Here's an image of it causing some disease on the native tree Miro near Rotorua. And interestingly, this Phytophthora doesn't seem to cause much problem on the native trees in New Zealand. However, the introduced invasive pests, such as Pinus radiata, which has been planted widely through New Zealand, suffers serious diseases. Similarly, Phytophthora canovii is planted in Europe, causes massive problems on rhododendrons and other species in England and elsewhere. Now, many Phytophthora species are only discovered after they are found causing disease. And this sort of makes sense. You know, why would we go looking for these pathogens unless we saw them causing massive diseases? And here are some examples. Phytophthora totra, or Phytophthora podocarpi, formerly Phytophthora totrii, causes massive disease on Phytophthora totra in New Zealand. This species was only discovered after we saw the disease. Cowrie dieback. This is a truly heartbreaking and devastating disease, causes massive dieback, awful bleeding cankers. This species was new to science until we found the disease. And this sort of makes sense. Phytophthora pinifolia. This is probably one of the real most dreaded pathogens for the pine industry. It causes needle cast, but also causes these horrible stem and twig infections in pine. And this was identified in the early 2000s. And this is a really nasty, scary pathogen. It's in Chile and other parts of South America. So imagine, put yourself in the perspective of if you found a new Phytophthora species in your industry, what would you do? What impact would it have on you? I think a lot of people would like to think they could just forget about it and ignore it. But often you can't do that. For example, Phytophthora pluvialis causes a devastating disease in red needle cast called red needle cast in the pine industry in New Zealand. It's because of this disease I essentially got a job in New Zealand about 9 years ago, because the New Zealand government freaked out. They were terrified about the potential implications of this new pathogen that was new to science. And hence they employed me and my colleague Nari and a few other people, and they flew over to New Zealand to work on this important new pathogen. So new Phytophthora species can cause massive management problems for land managers. So imagine if you're a farmer. What are the disease impacts? What are the known hosts? What are the unknown hosts? How does it spread? How can I manage the disease? Can you use chemical controls? Some chemicals you need to know the species before you can apply it. So these are some really serious management challenges. And it's even worse for government often. What are the trade implications of this new Phytophthora species? Can you still sell your goods overseas? Do you need to notify your trading partners? If you don't notify your trading partners, are they going to think you're a bastard? What do you tell them? You know, often when you get a new species, you have very limited knowledge, and it makes things very challenging. Is it spread through your nurseries? Do you need to close your nurseries? Do you need to close the industries that get plants from your nurseries? Can we still access infected properties? You know, should you be quarantining large areas of your land? These are really serious management challenges for government. Does it affect recreation land, conservation land, indigenous land? These are all very serious issues that happen when we get a new Phytophthora species. And it's even worse because there are many undescribed Phytophthora species. Modeling suggests there could be up to 4 times more Phytophthora species than we know currently. So, you know, the more we look for them, we probably will find more of these species. Lots of them are not going to be big problems, but some of them will. So how do we manage this? It's a real challenge. So the aim of this work is to determine if the invasive risk of Phytophthora species can be determined based on measurable traits. So the idea is you get a new Phytophthora species. Can we go out there and measure characteristics of this new Phytophthora species in the environment or the laboratory? And can we work out the invasiveness of that species using a model? Because if we can work out if we can work out how invasive it is, maybe we can work out, you know, how much of a threat it's going to be to industry, to government, to trade, and so on. So that was the aim of this work. So in order to do this, we decided to collect a lot of traits on all of the known Phytophthora species. So for all of the published Phytophthora species, we got data on the ecosystems that these Phytophthora species are known to impact. So for example, are the known Phytophthora species in natural ecosystems, urban environments, agriculture, perennial environments, water? We collected all of the data on the known Phytophthora species for these traits. Similarly, we collected data on the dispersal mechanisms. For all the known Phytophthora species, are they spread through the air like Phytophthora amorum or Phytophthora pluvialis? So they spread through the soil like Cinnamomi or Ostracidrii? Are they water specialists? What are the microenvironments they infect? That, some Phytophthora infect stems, roots, leaves, stem, roots, and leaves, What are the combination of these interactions? And also, we collected information on the phylogeny of these Phytophthora species. Wouldn't it be wonderful if you could get a Phytophthora species, look at the phylogeny, see what it's related to, and get a quick idea of the potential impact of that Phytophthora species? We also collected physiological data, information on the growth rate. These are all traits that can be measured quickly and efficiently in the laboratory. And then we also collected a lot of information on the morphology of these Phytophthora species. So information on the sporangia, on the oogonia, the size of the reproductive structures. And we built a trait database. So all of the traits in the red circles are traits you can measure in the laboratory. And all the traits in the blue circle are traits that you can measure in the natural environment. And the idea is that if a government or a laboratory found a new Phytophthora species, relatively quickly they could go and measure all these variables. And then we're hoping that we could develop a model that could predict the invasiveness based on these measurable traits. So the idea is we wanted to determine the invasiveness of known Phytophthora species. Is it a really mean species? Does it infect many different plants like a ramorum? Does it spread rapidly like a ramorum or a cinnamomi? Maybe it's not so mean. There are some Phytophthora species that infect and kill less plants. They spread very slowly. Maybe they're limited by environmental variables. And there are some Phytophthora species which are probably not that mean. Remember, Phytophthora species are part of the natural environment. They probably serve a purpose in particular environments. They might kill off weedy plants, or they might help in the dispersal of host plant material. For example, Phytophthora canovae probably has an important natural function within the New Zealand natural environment. But in Britain, enemy number one. Well, an enemy. We also collected information. So we've got all of these wonderful traits for these Phytophthora species, all the known Phytophthora species. We've got all this information that you can measure in the laboratory, you can measure in the natural environment. Then for all of these Phytophthora species, all the published Phytophthora species, we wanted to determine how invasive and how damaging they are. So to do that, we did a lot of web scraping. So we obtained data on the published disease reports for all these Phytophthora species. And we got, 16 to 20,000 disease reports globally. We collected information from the United States Department of Agriculture, CABI, the Centre of Agriculture and Bioscience. Got a lot of data from New Zealand Forest Research, Manaki Whenua Land Care Research. We also got about nearly 13,000 disease reports from GeneBank, NCBI. We also scrolled about 500 manuscripts for disease records that are not in these global databases. And then we started to try and work out the best way to predict invasiveness for these species. And we came up with an approach where we used data, including the number of countries that a Phytophthora species is known to be found in, the rate of these Phytophthora species being found in different countries, the number of hosts that a Phytophthora species is known to infect, and the rate of identifying new hosts for these Phytophthora species. And then we started to make a lot of phylograms, produces heat maps, and then we were able to produce unified indices of invasiveness for these Phytophthora species. And this was an approach that was actually very effective in the end. We produced a lot of different Bayesian models. This is where we're going to use that awful word, machine learning algorithms. But essentially, we produced tree augmented networks, Bayesian networks, which is a wonderful approach for producing predictive models. The wonderful thing about Bayesian models is they can use continuous data, categorical data, expert opinion. It's a very flexible platform for producing data and for analyzing data. I'm happy to discuss this in more detail, but I won't stay too much on this at the moment. But I will also just clarify that we produced all these models and we used a range of different cross-validation approaches. And we produce this beautiful Bayesian network, which might not look like much, but this weird array of tables and figures, there's a hell of a lot of data and information behind this network that can be used for doing predictive analysis. And it's actually I think it's really exciting and really powerful once you get behind the nuts and bolts of it all. So you can see in the middle, we've got our wonderful invasiveness scores. So is it a very invasive, low invasive, moderate invasive, high invasive, very high invasive Phytophthora species? And then these are the traits that the model said were the most informative for predicting the invasiveness. So these includes things like the year since it was first described, information on the impacted ecosystem, the dispersal mechanism, the physiology, the morphology of the Phytophthora species, the microenvironment. And this is our Bayesian network. And this was very exciting. We were able to produce a model with all of our different cross-validation approaches that had a less than 1% accuracy rate, less than 1% error rate. So it was over 99% accurate at predicting the invasiveness of all the known published Phytophthora species based on their measurable traits. The analysis also is able to give us a sensitivity of the traits. So this tells us how good are the different traits for predicting invasiveness. So for example, knowing which subclade a Phytophthora species is by and large the most informative trait for determining the invasiveness of that species. And that makes sense. If a new species is related to a very pathogenic Phytophthora species, it's more likely to be pathogenic. If a new species is related to not very pathogenic Phytophthora species, it's less likely to be very pathogenic. The model, so all of the different traits vary in their predictive ability. We eliminated some of the less predictive traits in order to make a more informative and accurate model. Some of the traits were a bit redundant. So for example, phylogenetic subclade was a bit redundant to phylogenetic clade, so we removed those traits. But by and large, this was a wonderful outcome. We had a very low error rate. And what does this give us? What does this give us for management? So it really comes back to this model. If you get it, we've developed a system where you get a new Phytophthora species, you can go out there, measure all the traits, put it into our model, and you'll get an indice of invasiveness. Our model will help you predict how invasive your new Phytophthora species is. And I'd just like to mention that through Plant Health Australia, we've recently got funding to do a residential workshop at Murdoch University in November. So this will be on using Bayesian causal networks in biosecurity risk assessment. It's a free workshop. It'll be happening in November. So if you're interested, please feel free to contact me. I'd love to have your involvement. We'll be looking at things like polyphagous shot hole borer. We'll be looking at myrtle rust. Within the workshop, we aim to actually produce some causal networks, predictive networks for the management of myrtle rust and polyphagous shot hole borer. Bruce Marcotte, who's going to be managing the workshops, very generous with his time. He's also very keen at publishing. So we actually hope to produce some manuscripts as a result of these workshops on polyphagous shot hole borer and myrtle rust. Thank you very much. Do we have any questions? Yes. Yeah, so that's one of the aims that we want to do from this workshop. So how we envisage this is going to happen is that before the workshop, I'm going to collect all the data I can on myrtle rust and polyphagous shot hole borer. I'm going to make a causal network. And then the participants who come to the workshop, we're actually going to put this network up on the screen and we're going to elicit expert opinion. We're going to tweak the variables and we're going to try and make the most predictive, valuable model that we can. And the aims I think we'd like to get, we'd like to, ideally we'd like to be able to produce a model that can somewhat predict the risk of myrtle rust in different environments within Western Australia and within different plant species. That's one potential outcome. Another outcome would be, You can use these Bayesian networks to help optimize your surveillance design. And something similar with polyphagous shot hole borer. We're hoping to really make a model as a result of this network of using polyphagous shot hole borer, predicting the susceptibility of different hosts, optimizing the surveillance design, and so on. Very careful how I respond, but yes, I think there are lots of caveats there. I think rubbish in, rubbish out, you've got to be very careful how you define your parameters. But yes, I think there is scope for these kind of causal networks to be able to use them to make informed management decisions. That's definitely being done for other diseases. A similar approach to this was used, for example, for optimizing management of the Fitzgerald River National Park in the early 2000s, I think. Any other questions? Well, thank you very much. It was a real pleasure to give that presentation. So now I'd like to welcome our people for the panel discussion. So